Alright everyone, welcome back to the channel. Before we dive into today's fascinating, and maybe slightly surprising, little experiment, I want to take a quick moment to send out my very best wishes to each and every one of you watching. Seriously, wherever you are in the world, I hope you're feeling good, full of energy, and that you and your loved ones are staying happy and healthy. Taking care of ourselves is the most important thing. Now, get ready, because today we're going to play detective with plants. We're going to uncover a secret hidden inside every green leaf, and we're going to do it using something you might find in your medicine cabinet or cleaning supplies: alcohol. That's right, we're going to boil a leaf in alcohol and see what happens. It might sound strange, but trust me, the result is pretty amazing and teaches us something fundamental about how plants work. It's simple, safe if we do it carefully, and reveals the magic of the green world around us. So, picture a leaf. Any green leaf will do. Maybe you pluck one from a tree in your yard, or a houseplant, or even find one on a walk. It's green, right? That green color comes from a super important chemical inside the leaf cells called chlorophyll. Chlorophyll is absolutely vital for life on Earth because it's what allows plants to do photosynthesis. Photosynthesis is that incredible process where plants take sunlight, water from the soil, and carbon dioxide from the air, and they use the energy from the sun to turn those simple ingredients into sugar, food for the plant. And as a wonderful bonus for us, they release oxygen into the air while they're doing it. So, chlorophyll is like the plant's solar panel and food factory all rolled into one, and it just happens to be bright green. But here's the thing. Chlorophyll isn't the only chemical inside a leaf. There are others, like yellow xanthophylls and orange carotenes, which you see in carrots, for example, but the strong green chlorophyll usually masks them. So, how do we see those other colors? How do we really prove that chlorophyll is in there? That's where our alcohol experiment comes in. To do this safely at home, you'll need a few simple things. First, obviously, you need some green leaves. Fresh ones work best. Next, you need some rubbing alcohol, the kind you use for disinfecting cuts, usually called isopropyl alcohol. You'll also need a small pot or a heat-proof glass container, like a beaker if you have one, but a small jar will work too. Crucially, you need a larger pot to act as a water bath. We are not going to boil the alcohol directly over a flame or hot plate. That could be dangerous, because alcohol fumes are flammable. Instead, we use the safer water bath method. You'll also need some water, a pair of tweezers or tongs, a spoon, maybe some white paper towels or coffee filters, and possibly a source of heat like a stove or a hot plate. And always, safety first. Wear safety glasses if you have them. Do this in a well-ventilated area, maybe near an open window. Keep flammable things away, and definitely have an adult help if you're young. Boiling water and hot alcohol need respect. Alright, let's get started. First, take your larger pot and fill it about halfway with plain water. Put this pot on your heat source and bring the water to a gentle boil. While that water is heating up, take your smaller pot or heat-proof container and pour in enough rubbing alcohol to cover the leaf you want to test, maybe half a cup or so. Now, take your green leaf. It's a good idea to soften it up a bit first to help the alcohol get inside. You can do this by dipping it in just boiled water for about 30 seconds to a minute. Carefully take it out, use your tweezers or tongs, and then immediately place it into the container with the room temperature rubbing alcohol. Make sure the leaf is fully submerged. Now, here's the important safety step. Carefully place this smaller container inside the larger pot of boiling water. The boiling water surrounds the smaller container, heating it up indirectly. It's like a double boiler, often used for melting chocolate gently. This way, the alcohol inside the smaller container will get hot, very hot, and it will start to simmer or even boil gently, but it won't be exposed directly to the intense flame, significantly reducing the fire risk. Keep the larger pot simmering or gently boiling. Now, watch carefully. Almost immediately, you might notice something happening. The alcohol starts to change color. It begins to turn green. And not just a faint green, often a really vibrant, deep emerald green. This is incredible. What you're seeing is the chlorophyll being dissolved out of the leaf and into the alcohol. The heat speeds up this process. The alcohol acts like a solvent, breaking down the leaf tissues and pulling the chlorophyll molecules right out. Leave the leaf simmering in the hot alcohol bath for maybe 5 to 10 minutes. You'll see the alcohol getting greener and greener. Meanwhile, look at the leaf itself. It starts to change too. Its vibrant green color fades. It might turn a pale yellow or brownish or even a bleached white, depending on the leaf and how long you leave it in. It becomes limp and fragile. Why? Because its main color pigment, its solar engine, is being stripped away. After 5 to 10 minutes, very carefully remember, everything is hot. Use your tongs or tweezers to lift the leaf out of the hot green alcohol. Place it gently onto a white paper towel or a coffee filter. Look at it closely. What color is it now? Chances are it's lost almost all its green. You might see it's a pale yellow or maybe even whitish. It looks like a ghost of its former self. It might be quite delicate, almost see-through in places. This is your leaf without most of its chlorophyll. The green has literally been washed out into the alcohol. But sometimes, especially if you used a leaf that was starting to turn in autumn, you might see hints of yellow or orange remaining on the leaf itself. These are those other pigments, the xanthophylls, yellow, and carotenes, orange, that were always there, hidden by the dominant green chlorophyll. Boiling in alcohol primarily removes the chlorophyll, often leaving these other colors more visible on the leaf itself, or sometimes they dissolve into the alcohol too, making the green liquid look more olive or brownish. Now, look at the alcohol in your small container. It's transformed. It's no longer clear. It's a deep, rich green. This green liquid is essentially a solution of chlorophyll dissolved in alcohol. You've successfully extracted the pigment. You can even try dipping a piece of white paper towel or a coffee filter into this green alcohol. When you pull it out and let it dry, you'll have a piece of paper-stained green with pure chlorophyll. This is a basic form of what scientists call extraction, pulling out a specific chemical from a complex mixture, like a leaf, using a solvent, like alcohol. It's a fundamental technique in chemistry and biology labs. But wait, there's more we can learn. Remember how we talked about photosynthesis? Chlorophyll helps make food, sugar, for the plant, and plants often store some of this food as starch, especially in leaves. We can actually test our boiled leaf to see if it had starch in it before we boiled it, even though the chlorophyll is gone. Here's how. After you take the leaf out of the alcohol and it's cooled down a bit, you need to wash it gently in warm water. This rinses off any excess alcohol and softens it again. Then, lay it flat on that white surface. Now, get some iodine solution, the kind you might have in a first aid kit, usually diluted tincture of iodine, or you can use Lugol's iodine if you have it. Iodine is a classic test for starch. Carefully drop a little iodine onto the leaf, or better yet, place the leaf into a small dish with a bit of iodine solution for a minute or two. Watch what happens. If the leaf did contain starch before you boiled it, the areas where the starch was present will turn a very dark blue-black color. It's a dramatic change. This happens because iodine molecules fit inside the coiled structure of starch molecules, forming a complex that looks dark blue or black. If there's no starch, the iodine might just stain the leaf a lighter brownish color. Seeing those dark patches tells you that this leaf was actively making and storing food via photosynthesis. It's proof that the chlorophyll wasn't just for show. It was doing its job. Think about what this experiment shows us. First, it visually proves that the green color in leaves is caused by a specific chemical, chlorophyll, that can be physically removed. The leaf without chlorophyll looks completely different. Second, it reveals that leaves often contain other colored pigments, yellows, oranges, that become visible once the green chlorophyll is gone. This is exactly what happens in the fall. As days get shorter and cooler, trees stop producing chlorophyll. The existing chlorophyll breaks down and isn't replaced. Without the dominant green pigment, the yellows and oranges that were always there can finally shine through, giving us those beautiful autumn colors before the leaves eventually fall. Our boiling experiment just speeds up that chlorophyll removal process dramatically. Third, by testing for starch with iodine, we get direct evidence linking the presence of chlorophyll, even though we removed it, to the process of food production inside the leaf. Where there was chlorophyll actively working, there was likely starch being made and stored. It connects the green color directly to the leaf's function. So, what happened to the leaf itself? Well, boiling it first in water and then in hot alcohol is pretty tough treatment. The heat breaks down the leaf's structure. The alcohol dissolves not just the chlorophyll, but also fats, waxes, and other components. What you're left with is often very fragile. The basic skeleton of the leaf, made mostly of cellulose and maybe some veins. It might tear easily. You've essentially created a very simple, somewhat bleached leaf skeleton. It's not as intricate as the skeletons you might make by carefully rotting away the soft tissue over weeks, but it shows how the chlorophyll and other soluble parts were crucial for the leaf's color and much of its substance. This simple experiment, boiling a leaf in alcohol, opens a window into the hidden chemistry of plants. It makes the invisible process of photosynthesis a little more tangible. You literally see the green engine of life being pulled out of the leaf. You see the other colors it was hiding, and you can test the evidence of its food-making work. It's a powerful reminder that the green world around us is constantly busy, powered by sunlight, capturing carbon dioxide, and releasing the oxygen we breathe, all thanks to this amazing green molecule. The next time you see a green leaf, you'll know that inside is a complex chemical factory, and that vibrant green is the sign of its active, life-sustaining work. Thanks so much for joining me in this exploration of the secret life of leaves. I hope you found it as fascinating as I do. If you try this experiment, be super careful with the heat and the alcohol, and have fun being a plant detective. Wishing you all a wonderful day filled with curiosity and appreciation for the natural world. Stay healthy, stay happy, and I'll see you in the next video. Take care.